Welcome to a fascinating journey where the ancient world of fungi meets cutting-edge quantum physics. Today, we'll explore how researchers at the University of Ghent in Belgium have harnessed the power of common oyster mushrooms to create a revolutionary medical imaging technology that could transform healthcare as we know it. These biodegradable quantum dots represent not just a scientific breakthrough, but a perfect example of how nature's solutions often surpass our synthetic attempts. Join me as we delve into this remarkable convergence of mycology and quantum physics that's creating safer, more effective medical imaging while potentially solving environmental challenges across multiple industries. In February 2023, researchers at the University of Ghent published a groundbreaking study in advanced sustainable systems that has sent ripples through both the medical and physics communities. They successfully engineered biodegradable quantum dots using enzymes extracted from the common oyster mushroom, scientifically known as Pleurotus austriatus. This development represents a critical solution to one of medical imaging's most persistent problems: the toxicity and environmental persistence of traditional quantum dots. Conventional quantum dots, while incredibly useful for their light-emitting properties, typically contain heavy metals like cadmium and lead. These toxic components have limited their clinical applications despite their extraordinary optical properties. The Belgian research team, led by Dr. Elke van der Meers, discovered that specific lacase enzymes found in oyster mushrooms can catalyze the formation of carbon-based quantum dots that perform just as well as metal-based versions, but without any of the associated toxicity. The innovation lies in the extraction method developed by the research team. They use a proprietary cold pressure technique that preserves the structural integrity of the enzymes while maximizing yield. The resulting quantum dots are incredibly tiny, measuring just 2 to 5 nanometers in diameter, which is approximately 50,000 times thinner than a human hair. What makes these fungal-derived quantum dots truly remarkable is their complex carbon lattice structure. This structure creates quantum confinement effects similar to those found in semiconductor materials but is achieved through entirely biological mechanisms. As Dr. van der Meers explains, nature has been engineering nanostructures for billions of years. The lacase enzymes in oyster mushrooms have evolved to break down some of the most recalcitrant materials on Earth. We've essentially redirected this ancient biochemical pathway toward creating precision nanoparticles. The discovery came when researchers observed how these fungi naturally process lignin, a complex polymer found in wood, breaking it down into nanostructures with remarkable optical properties. These fungal quantum dots exhibit remarkable photoluminescence across the visible spectrum, with quantum yields exceeding 60% in optimal conditions. This makes them competitive with many commercial semiconductor quantum dots currently in use. One of the most valuable features is that their emission wavelength can be tuned by adjusting the enzymatic reaction conditions. This allows for customized imaging applications without introducing new toxic elements. The performance metrics are particularly impressive considering these are entirely biological in origin. When excited by specific wavelengths of light, these quantum dots emit precisely controlled frequencies of light that can be detected with extreme sensitivity by medical imaging equipment. This property is crucial for detailed medical diagnostics, especially when trying to visualize microscopic structures within the human body. What makes this discovery particularly remarkable is the rapid transition from laboratory to clinical application. The fungi-derived quantum dots have already been used in a small pilot study at University Hospital Brussels to image lymphatic vessels in patients with post-surgical lymphedema, a common complication following cancer treatment. Dr. Peter van Hove, a radiologist involved in the trials, noted that the resolution we achieved exceeded our expectations. But more importantly, we observed complete biodegradation of the imaging agent within 72 hours, compared to conventional quantum dots that can persist in tissues for months or years. This represents a paradigm shift in how we approach contrast agents for medical imaging. This rapid biodegradation is crucial for patient safety and represents a significant advancement over current technology. The body's natural enzymes break down the fungal quantum dots into simple sugars and amino acids that enter normal metabolic pathways, mimicking the natural process by which fungi themselves recycle complex organic materials in forest ecosystems. The clinical implications extend far beyond lymphatic imaging. Preliminary research suggests that these quantum dots can be functionalized with targeting molecules to identify specific cancer cells, potentially enabling earlier detection of microscopic tumors. Unlike conventional imaging agents, the fungal quantum dots can penetrate the blood-brain barrier when administered intranasally, opening new possibilities for neurological applications. Neurologist Dr. Myco-Quantum has begun exploring the technology for tracking amyloid plaque formation in early-stage Alzheimer's patients. The non-toxicity profile means we can potentially perform serial imaging studies that would be ethically questionable with conventional quantum dots, Dr. Devert explains. This allows us to track disease progression with unprecedented temporal resolution. This capability could transform our understanding and treatment of neurodegenerative diseases by allowing safe, repeated imaging that was previously impossible due to toxicity concerns. This research highlights the immense value of cross-disciplinary collaboration in scientific breakthroughs. Dr. van der Meersch, who was initially trained as a mycologist, partnered with quantum physicist Dr. Julian Kessler after a chance meeting at an interdisciplinary conference in 2020. I was studying bioluminescent fungi and Julian was working on quantum dots for solar cells, van der Meersch recalls. We realized the enzymatic processes in certain mushrooms might solve the toxicity problems in his field. What began as a casual conversation over coffee has evolved into a complete reimagining of how we produce nanomaterials. This serendipitous connection between experts from seemingly unrelated fields demonstrates how innovation often occurs at the intersection of diverse disciplines. By combining their specialized knowledge, these researchers were able to see connections and possibilities that might have remained hidden within the silos of their respective fields. The implications of this technology extend well beyond medicine. Environmental scientists at Zurich are now investigating whether similar fungal enzymes could help remediate soil contaminated with heavy metals from electronic waste. This is becoming an increasingly urgent global concern as discarded devices containing conventional quantum dots accumulate in landfills. The same enzymatic pathways that create the quantum dots can potentially be reversed to break down toxic nanoparticles in the environment. This application could provide a biological solution to a growing environmental crisis, using nature's own mechanisms to clean up human-made pollution. The potential for using fungi to remediate contaminated soils represents a sustainable approach to environmental cleanup that avoids the need for harsh chemicals or energy-intensive processes. In agriculture, researchers at Wageningen University are exploring these biodegradable quantum dots as sensors for plant pathogens, potentially allowing farmers to detect disease outbreaks before visible symptoms appear. This early detection system could revolutionize how we protect crops, enabling precision agriculture interventions that minimize chemical inputs while maximizing crop protection. By applying the quantum dots to plant tissues, researchers can create imaging systems that detect subtle changes in plant physiology that indicate the presence of pathogens. When a pathogen begins to affect a plant, the quantum dots can be designed to change their emission properties, creating an early warning system visible with specialized imaging equipment. This could dramatically reduce crop losses and decrease the need for preventative pesticide applications, supporting more sustainable farming practices. Perhaps most surprisingly, materials scientists at MIT have begun investigating how the self-assembly properties of these fungal quantum dots might inform new approaches to sustainable electronics manufacturing. Dr. Samira Patel of MIT's Biomimetic Materials Laboratory notes, these biological systems achieve at room temperature and ambient pressure what we typically require energy-intensive clean rooms and harsh chemicals to accomplish. This biomimetic approach to electronics manufacturing could significantly reduce the environmental impact of producing electronic components. By studying how the fungal enzymes create precise nanostructures under mild conditions, researchers hope to develop new manufacturing processes that require less energy and fewer toxic chemicals. This could transform electronics production from one of the most environmentally problematic industries into one that follows nature's sustainable design principles. Beyond the scientific and environmental benefits, there are compelling economic advantages to this technology. The production cost of these biodegradable quantum dots is approximately 40% lower than traditional semiconductor-based alternatives. This cost efficiency stems primarily from the raw materials, oyster mushrooms, which can be cultivated using agricultural waste products such as coffee grounds and sawdust. This circular economy approach has attracted significant attention from sustainable technology investors. Belgian biotech startup Myco-Quantum has already secured €12.5 million in funding to scale production. The company projects that fungi-derived quantum dots could capture 30% of the medical imaging contrast agent market within 5 years. This represents not just a scientific breakthrough but a commercially viable alternative to current technologies, with the potential to disrupt multiple industries while providing environmental benefits. Technology forecaster Dr. Sophia Yamamoto, who was not involved in the research, suggests we're witnessing the birth of an entirely new field, mycoelectronics. This intersection of fungal biology and quantum physics could transform multiple industries, from medical imaging to environmental sensing and beyond. The elegant simplicity of using organisms that naturally decompose matter to create cutting-edge nanomaterials represents circular economy principles at their finest. This emerging field represents a fundamental shift in how we approach technology development. Rather than forcing synthetic materials to perform functions they weren't designed for, microelectronics leverages biological systems that have been perfected through billions of years of evolution. As researchers continue to explore the capabilities of these biodegradable quantum dots, we're likely to see applications that haven't even been conceived yet, spanning healthcare, environmental monitoring, sustainable computing, and beyond. As we reflect on this remarkable convergence of biology and physics, one thing becomes clear. Sometimes the most advanced technologies have their roots in the most ancient life forms on our planet. The oyster mushroom, a humble organism that has been decomposing fallen trees for millennia, now stands at the frontier of quantum nanotechnology. This reminds us that innovation often lies not in creating entirely new materials, but in understanding and repurposing the sophisticated molecular machinery that evolution has perfected over billions of years. As Dr. van der Meersch humbly notes, in the end, we didn't invent anything new. We simply learned to speak the biochemical language that fungi have been using for eons. The quantum properties were there all along. We just needed to look in the right places and ask the right questions. As we face growing challenges related to resource scarcity and environmental contamination, such biomimetic approaches offer pathways to technologies that are inherently aligned with natural cycles, potentially transforming how we approach innovation across all fields of science and technology. From the laboratories of Belgium to potential applications worldwide, fungal quantum dots represent a perfect harmony between nature's wisdom and human innovation. By leveraging the enzymatic capabilities of oyster mushrooms, scientists have created imaging technology that is safer for patients, better for the environment, and more economical to produce. This breakthrough reminds us that sustainable solutions often exist within the natural world, waiting to be discovered. As we continue to face complex challenges in healthcare, environmental remediation, and technological development, the humble mushroom offers a powerful lesson. Sometimes looking backward to biological systems that have evolved over billions of years can propel us forward more effectively than synthetic approaches alone. The convergence of mycology and quantum physics doesn't just solve existing problems. It opens entirely new frontiers of possibility across multiple disciplines. As fungal quantum dots move from research to widespread application, they represent not just a technological advancement, but a fundamental shift in how we approach innovation, working with nature rather than against it to create technologies that are inherently sustainable, biodegradable, and aligned with the circular principles that govern our natural world. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.